Good morning, everyone. Good afternoon, good evening, depending on where you are in the world. Thank you guys for joining us today for this webinar. I'm going to give it a couple more seconds till we probably are a minute past, just to let people join in, and then we'll get started. So just hang tight as the people get settled in and join the webinar. Alrighty, so it looks like those numbers are stabilizing. So good morning, everyone, again. My name's Avi. I'm based out of Boston. I work for the marketing team here at CELLINK, and it's a great pleasure to welcome everyone to this exciting webinar that we've got planned for you guys today. We are very fortunate to have Dr. Charles Tsai, who is the CEO at CYTENA Bioprocess Solutions, and he's going to be giving us a walkthrough on the C.BIRD and really discuss how this product has the potential to revolutionize small-scale cell culture. So before we get started, I'd like to just go over some housekeeping rules. So everyone has entered the webinar on mute, but we do want to hear from you. So if you have any questions, you'll see a little questions tab at the bottom of your control panel. We will be monitoring that throughout the presentation, so if anything urgent comes up, feel free to put that in the questions chat. If not, there is a question and answer segment at the end of this webinar, and we will definitely be going over all the questions with Charles, so you will have time to pick his brain. And in addition to that, we do have a couple of polls today in this webinar, and so what you'll see on your screen is questions with a couple of options. Please answer those questions on the screen, not in the question box. So with that, I will turn it over to you, Charles.
Okay, so hi everyone. Thank you for attending the webinar today. And I'm Charles, and I'm very happy to be here and give an introduction about C.BIRD technology, which will be the next revolution of the cell culture. And there will be a Q&A session in the end of the presentation. As Abby said, you can anytime to write down your questions, and we will picking up in the end of the presentation. And so yeah, let's just get started. I believe most of the scientists understand the importance of the mixing for cell culture. For traditional cell culture, cells are limited to a 2D growing space. Oxygen comes from the gas-liquid interface, which is at a far distance to the cells. So all the waste metabolics that cells produce are also concentrated around the cells. This makes an ideal cell culture environment. So if we enable the mixing culture, suspension cell line can have a 3D growing space. All the cells can have equally distribution of the oxygen or nutrition. Even for the adherent cell line, it can also refresh the medium surrounding the cells as well. So for the mixing culture, is quite important for a cell culture. And there's a poll we would like to know what kind of cells you are working on currently. Is it a suspension type or adherent type of cells? Okay, I will wait a little bit to wait until everyone vote. Okay. Okay, let's keep going. So here is the cell line development process published by some well-known pharmaceuticals company. Cell line development process typically starts from a single cell culture in the 96 well plate and continuously scaling up to 24 well plate or a check-in place or a 24 deep well place in the later stage of a bioreactor. As you might have already noticed that the mixing of the cell culture only begins from the check-in place or 24 deep well plate in a later stage. And however, in the first two stages, 96 or 24 well plates current technology in the market do not enable mixing culture. Therefore, pharma companies can only do steady culture, which is not ideal cell culture method. This is where C.BIRD plays a revolutionizing role here. C.BIRD wants to fulfill this gap to enable the suspension mixing culture in both 96 well and 24 well plates. And C.BIRD can offer a 3D suspension environment for small-scale cell culture based in the 96 or 24 well plate, which is difficult to achieve by checking culture or any other state-of-the-art currently. So one C.BIRD system consists of docking station and 3 control units, which can operate 3 96 or 3 24 well plates. The C.BIRD working principle is quite simple. There's a C.BIRD lead, which can fit with a standard 96 well plate, and there are 96 tubes, which can be inserted into all the wells of the well plates. And the C.BIRD control unit can be docked right on the top of C.BIRD lead to pumping up and down the medium in all the wells, somewhat like a multi-pipetting system. So just pumping up and down of the cell culture suspension inside all the wells. So like this, basically all the wells were working like this, so like a multi-pipetting system. As shown in the video on the bottom right of the slide here, we have placed a camera at the bottom of the well while C.BIRD is operating. You can see that CHOK1 cells are being pumping up and down in the well. Okay, and I will now share some data of the C.BIRD culture in the following slides. This will help you understand the difference that C.BIRD culture can bring to your cell line development process. So before showing the data, I would like to point out that the C.BIRD enable customizable mixing option. While mixing is generally good for cell growth, but too much mixing may be damaged for very low cell density. Hence, at the low cell density culture, or for example, from a single cell level to 1,000 cells per milliliter, in this range, we usually recommend periodic mixing. So for example, every 12 hours or every 6 hours, you can mix in for 10 cycles. You can have this setting in a C.BIRD culture. And from a higher than cell density level, we recommend continuously mixing of the cell culture for most optimal growth. The mixing condition can be tailored to your needs and also a different cell type. So just like shaker, you can set in different shaking RPM as well. And there's a pool as well here. We would like to know what kind of cell concentration you're normally working on. Okay, so let's go into the data part. So in this experiment, we look at the cell growth and the protein yield for the CHOK1 cell that produce a HER2 antibody used for the treatment of the breast cancer. We compared C.BIRD to steady culture in the 96 well culture plates and took the endpoint measurements. So the case 1 here, we seeded 500,000 cells per milliliter and the culture for 5 days. And the case 2, we seeded 1.5 million cells per milliliter and the culture for 3 days. As you can see from the chart on the right here, C.BIRD consistently produced much higher cell density and the antibody protein yield compared to steady culture in both case 1 and case 2. C.BIRD also produced a higher QP value here compared to steady culture. This experiment showing that a C.BIRD culture provide more optimal cell growth for higher cell density and the protein yield. So in both cases. And for this case, a C.BIRD mixing setting is 25 seconds and continuously mixing. So for entire 3 to 5 days. Okay, and the second data set here is generated by our customers. So we don't know the titer, exact titer, which titer is producing, but it's showcase one cell as well. Let's see these cells at 3 different cell density. For these 3 different cell density and marked here. And compare C.BIRD also with steady culture for cell viability and the density after 3 days of culture. So it's also an endpoint measurement. And so at the low cell density, C.BIRD do not differ greatly from steady culture in terms of cell viability. However, at higher cell density, seeding density, C.BIRD enables much higher cell growth up to 5 million cells per milliliter with 98% cell viability here. Steady culture only has 1.37 million cells per milliliter with only 84 viability. So it's showing that in a higher cell density, C.BIRD can always push the cell line growth to a higher number. And experiments shows that the C.BIRD can strengthen the growth potential of the cell culture when steady culture reaches its limit. And C.BIRD can bring it further more to a later stage. And the C.BIRD can push it to a more higher cell density and keep in a relative high viability as well. And we also did a correlation assay to see how well C.BIRD compared to the shaker condition. We compared C.BIRD and the steady culture to 125 milliliter shaking flask with around 30 milliliter cell suspension. And we put in a shaker for this shaking flask with 150 RPM, apical diameter is 90 millimeters. And we measure the cell density every day for continuously for 5 days. And you can see that the cell density of the C.BIRD and the shake flask group are similar for cell density and also for the viability as well. But steady group can only remain at a lower level. So it cannot be being higher after around 1.5 million cells per milliliter. This shows that cell growing in C.BIRD condition mimics the growth rate of the cell grown in the shaker flask. The same goes for the doubling time and also the titer of the protein yield after 5 days. The data shows us that a C.BIRD culture condition is much more representative of the later stage shaker flask conditions. This is extremely important for early selection of the cell clones, which saves time and cost for cell line development process. Okay, and we also measure the oxygen value at the bottom of the well. So the data shows that the C.BIRD condition increase the oxygen level at the bottom of the well compared to steady culture conditions. So as you can see this figure, the gray curve here is just pure medium without any cells inside. And the dark blue curve here is steady cell culture. So after 2 hours of a culture at the bottom of the well, an oxygen level dropping down very quick. And C.BIRD culture can also still remain a relative high number here. And this is important in preventing hypoxia in cells. which may be important for some researchers as well. Okay, so here we can also show that C.BIRD has no cross-contamination issues. One of our customers tested this by intentionally only adding cells to row A, C, E, and G here, and continuously culture for 7 days, and also using C.BIRD to mixing continuously for 7 days. And after 7 days, they only see cells in row still in the ACEG line, but no cells to row BDFH line here. And so it's showing like the cells won't, cross, go into another wells. And at the same time, they are measure the titer production, as shows in the figure. C.BIRD performs a higher titer than the steady culture as well. And just to mention here, you may see some patterns here. That is because in the end, they're using centrifuge to make cell sediment at the bottom of the well, so that there's some pattern happens. It's not generated by C.BIRD. And these slides show an example that C.BIRD can also have benefit for the low cell density. So in this experiment, our customers seeded single cell into 6 plates and separate to C.BIRD group and also steady cell culture group. 3 plates for each group. And then from day 1 to day 10, C.BIRD group also remains steady culture because our customer wants to take picture of the single cell clone. Also, FDA requests this single cell cloning of improvement. And so from day 11 to day 14 in the C.BIRD group, we start to using C.BIRD mixing for 3 days. And for the steady culture group, for the first 14 days, it remains static for the first 14 days. And then we pick up the top 12 clones and measure the titer. And as you can see from the figure here, C.BIRD has better performance in average. And and then we're also continuously scaling up these top 12 clones to the shaking 6 well plate, and then measure the titer again. And C.BIRD group still has a better performance than a steady culture group. So one thing need to be noticed here, we only use periodic culture in this experiment because of a low cell density. If the mixing is too strong, there might be some negative impact to the cells, but it might be different from a cells line to cell, different cell lines. And this data is also generated by our customers. Okay, so one set of a C.BIRD microbioreactor includes 1 docking station and also 3 control units. And the whole device is very compact and able to put into an incubator. As mentioned, the mixing rate here is adjustable for different cell lines, even setting as a periodic mixing. And also, only 1 single USB cable is required to connect into a laptop for power supply and data transfer. And 1 laptop can connect up to 3 to 5 sets. So that means 9 to 15 control unit of the C.BIRD. And the current consumable is fitted with open-top cell culture plate, as it's special designed to prevent evaporation. And 24 well plates, these will be available soon in October. And yeah, so today we only focus on the cell line development market, but there are potential to use in different applications as well. We have been asking from different fields, for example, yeast culture, stem cell culture, or cancer cell culture, spheroid research as well as microbiome or yeast culture. So basically, we are not limited our application in the cell line development. So if you are in general interested in C.BIRD, please contact us, and we can discuss in different applications as well. So we are happy to discuss different possibilities. So also we have a last call here to know about what kind of research you are currently doing, and we are also very interested in our audience. is working on. Okay. Okay, so basically that's my presentation for today. So thank you, everyone. So I think here will be the Q&A session. Abby probably can take over from here.
Yeah, thank you so much, Charles. That was truly an informative presentation, and I think it's great to see the massive potential that the C.BIRD has in terms of small-scale cell culture. So we do have a couple of questions that have come in already, and folks who are listening, feel free to keep asking questions. So Charles, could you just quickly talk about the fact of, did you observe any evaporation issues during the C.BIRD mixing culture? So whilst you have the cells in mixing culture, were there any evaporation issues?
Yeah, probably as you can imagine, like mixing generally can increase evaporations. So that's the reason I just mentioned in the previous slides that we choose the append of cell culture plate currently to come to our C.BIRD lead. That is because the append of cell culture plate, they have a special design to prevent the evaporation, which you can add some PBS or some DI water in a mold area. I mean, the space in between the wells. So there's some kind of chamber design, and you can add some DI water so that you can increase the humidity surrounding our cell culture. So for this, we have testing for this purpose to culture continuously for up to 1 to 2 weeks. The evaporation issues can be limited. Okay.
Wonderful. And then another question that has come in is, how long could the C.BIRD operate inside the incubator?
Yeah, so basically it won't have the limitation, but the limitation also because of the evaporation I just mentioned previously. But in the 24-hour pace should be fine as well because there's more medium inside. But for 96-hour plate in general, as I mentioned, 1 to 2 weeks should be fine. And for 24-hour plates can be longer. Okay.
Gotcha. Okay. Could you help answer this question? Is there any option for measuring pH, CO2 or O2 concentrations and pressures?
Yes. So basically, in this generation of C.BIRD, it's only with mixing function, but it's very good questions that that is in our next development plan. So we will have a pH value and also the dissolved oxygen value being measured with the system for our next generation of the device. So as you can probably imagine, like for the shaking culture, it's probably hard to integrate this kind of optical sensors. But for our stable mixing culture, it's easily to integrate this sensors technology inside our system.
Okay, I think that's extremely exciting, and I think you rightly point out that those would be interesting parameters for people to monitor, especially within mixing culture. Alrighty, so I will give it a couple more seconds, but we have gone through the questions that have come in. If there aren't any other questions, what we can do is we can start to wrap things up. But folks, feel free to keep asking questions. Charles, can you just quickly clarify? So one of the pictures showcased 3 C.BIRD devices stacked on top of each other? Is that part of like the scalability of the system, or can people use just one, or what are the options there?
I understand some of the people want to using, for example, like 15 plates in the cell line development. So our system do offer this possibility. So if you purchase more than one set of the C.BIRD, and you can still connecting them directly. We will offer a hub, and you can connecting them directly to a hub that you can still control all of them by a laptop. A single laptop should be offer the power supply and also the data transfer.
Okay, great. And we did get one last question come in, and I think we can wrap things up after this. How easy is it to connect the plates to the C.BIRD? Or just in general, could you help clarify the process of connecting a plate to the C.BIRD control unit?
Yeah, so maybe I back to the picture previously. So you can see here, so basically the lead here, you can, without any techniques here, you can just put it on a standard web page. It doesn't require any kind of docking or any kind of techniques to put it on. So just like a standard lead. And when you put it, as I mentioned, there are 96 tubes that can be inserted directly into the tubes here. And then the docking station here, the control box up here, that it require a connector from this part and to this port. And you will hear a click sound once you put it on. So the whole things become very simple. So you just need these 2 steps, and you can put the entire sandwich units onto our docking station, and it can be operating. So our procedure remain quite simple.
Perfect. Yeah, it does look like a fairly straightforward and intuitive process. Okay, with that, I'd really like to thank everyone for joining this webinar. We hope that you found it useful. Thank you, Charles, for your time and your expertise. If you are interested in attending more webinars, we are constantly keeping our calendars up to date. You will find the most recent information on our website. So that's www.stellink.com/events. 2 webinars which are coming up, which may be of interest, is we're taking a dive deep into fresh printing. And so we're going to have a live demonstration on our BioX system coming up on the 7th of October. And then later on this month, on the 22nd, we're going to be taking a good look at 3D cell culture and touch a little bit upon some things that Charles spoke about at the end in terms of application areas. So we'll be looking at spheroids and how they are becoming an important part of the life sciences. So with that, I would like to thank everyone again for taking the time to join this webinar. If you are interested in seeing some of these products in your own lab, we are offering demonstrations, and we are figuring out the best ways to make those demonstrations possible, whether they're in person or virtual. We have all the capabilities available, so please don't hesitate to reach out to us to schedule a demo or a discussion. So we look forward to hearing from you, and with that, I'd like to wrap things up, and thank you everyone for taking the time, and thank you again, Charles.
Thank you very much for everyone